scientist, and I'm going on a crime scene. I've just been called. Okay, down the hall. I think it's here. Jerome, that's the door. Open up. Oh, there's a victim. Quick. Oh, there's been struggle.
This presentation is about a technique used for DNA profiling in forensic science. The technique is called polymerase chain reaction, or PCR. DNA profiling is used to identify an individual based on the DNA left at a crime scene. What's so powerful about PCR is that it allows you to analyze the DNA even if there's very, very little of it at the crime scene. Now, to understand this technique, PCR, we first need to review what DNA is. DNA stands for deoxyribonucleic acid. It's the genetic material that is found in all cells of your body. It defines you as part of the human species, and most importantly, defines you as a unique individual. DNA is made of building blocks called nucleotides, of which there are 4, hence the 4-letter code you might have heard of. These 4 letters are A, G, C, and T, and I will explain that in the next slide. This is the structure of a nucleotide. It's made of 3 components. The first component is a sugar. This is a 5-membered carbon and oxygen ring. The second component attached to the sugar is a phosphate. And the third component, circled here in blue, is the nitrogenous base. The nitrogenous base could be a double ring of carbon-nitrogen as shown here, but could also be a single ring. There are 4 different nitrogenous bases, 1 for each nucleotide. The 4 bases are called A, G, T, and C, and those letters are given as names to the nucleotides as well. A stands for adenine, the adenine base, G, guanine, guanine base, T, thymine, thymine base, and C, cytosine, the cytosine base. On the left, you have a simplified structure of a nucleotide. S is for sugar, P is for phosphate, and the C is for cytosine, one of the 4 nitrogenous bases. Now, nucleotides form together a chain or a strand called nucleic acid, and this is shown in the middle. Circled are the individual nucleotides, and they form a backbone of phosphate sugar, phosphate sugar, phosphate sugar, etc. Now, in DNA, you have 2 strands. This is a second strand. Both strands are said to be anti-parallel to one another, as you can see from the orientation of the sugars, and they're held together by hydrogen bonds between bases, opposite bases of the 2 strands. These hydrogen bonds are relatively weak, and so DNA can easily be melted. That is, the 2 strands can easily be separated by simply boiling the DNA molecule. This is an important concept to understand PCR that we will explain a little bit later. Here again is shown the DNA molecule made of 2 strands that are running in anti-parallel fashion. And in the middle, they're held by the bases that form hydrogen bonds with one another. As you can see, there is a specific pairing happening. The A base always pairs with a T base, and the G base always pairs with a C base. This is called the base pair rule, or complementary base pairing. So to recapitulate, DNA is made of two strands of nucleic acids that are anti-parallel and complementary. In solution and in the cell, DNA forms a beautiful molecule called a double helix. That is, the two backbones of the two strands twist around one another to form a helix, and the bases that pair with one another form the rungs of a ladder, as shown here. Okay, we're now ready to explain what PCR is. PCR, or polymerase chain reaction, is an amplification reaction that allows you to amplify the amount of DNA that is left at a crime scene. You might start with minute amounts, nanogram amounts of DNA, and by the end of the reaction, you'll end up with microgram amounts. So you essentially amplify 1,000-fold. Or you start with one copy of DNA at the crime scene and you end up with millions of copies. And then once you have amplified the DNA, you can then analyze it for DNA profiling. PCR is a repeated cycle of 3 steps, and you repeat those cycles 20 to 30 times. The steps are: the first one, denaturation, that is done at 100 degrees. This is the temperature that melts DNA or separates the two strands in DNA. The second step is called annealing, that's done at a lower temperature between 45 and 65 degrees and involves primers that will delimit the area of DNA that will be amplified. And the third step, extension, is a synthesis reaction that is done at 72 degrees. In this reaction, DNA is amplified, that is, new DNA is synthesized, and this requires an enzyme called Taq polymerase. Our own enzymes would not be able to do this. They would be denatured at those temperatures. What's so special about Taq polymerase, it was identified first in bacteria that are thermoresistant and are capable of surviving those high temperatures. PCR is done in a special machine called a thermocycler that rapidly increases and decreases the temperatures of the 3 cycles, of the 3 steps in the cycle. Shown here is a diagram of the 3 steps of a PCR cycle. But before I begin, let's just look at the top. At the top, we have a DNA molecule that will be amplified. We have the horizontal blue lines, which represent the backbone of the 2 strands. The letters are the nitrogenous bases that are held together, that are holding with one another of the 2 strands by these short vertical lines that represent the hydrogen bonds. In the first step, denaturation, the 2 strands are separated or melted. They are now called template strands. They will be template for the synthesis of daughter strands. In the second step, annealing, primer 1 and primer 2 are added. These are the short pink sequences. These are also called forward and reverse, and they anneal, they hybridize with their bases to specific regions of the template DNA. They in fact delimit the area that will be amplified. They represent the extremities of that region that will be amplified. In the last step, extension, Taq polymerase, which is not shown in the diagram, extends the sequence from the primers onwards and forms 2 new daughter strands that are in dark blue. And if you notice, the daughter strands will have sequences that are complementary to the template strand by the bases. So the Taq polymerase essentially adds nucleotide by nucleotide of the daughter strand based on the sequence of the template strand. This is a schematic representation of the amplification achieved by PCR. On the left is labeled the template DNA. That might be the DNA found at a crime scene. PCR will just amplify a section of that template DNA called the target. After 1 cycle of PCR, there would be 2 copies of the target. After 2 cycles, there would be double, 4 copies. After 3 cycles, 8, etc. So theoretically, there's an unlimited exponential amplification that is possible. Now, for a given set of primers, the amplification of an individual's DNA will give a target sequence length of maybe 100 nucleotides. If you use the same set of primers to amplify the target sequence of another individual's DNA, you might get a target sequence of 50 nucleotides. And that is the basis of DNA profiling. DNA profiling is really based on the fact that individuals have unique target sequence lengths. It's still possible, though, theoretically, that two individuals will have the same target sequence lengths after PCR using a given set of primers. And that's why DNA profiling is usually done with 13 or more different sets of primers in order to generate 13 or more different target sequences. Now, the probability using those 13 or more different sets of primers of having two individuals with the same target sequence length is very low, 10 to the minus 20. Essentially, very, very unlikely.
Today I'm all ready, dressed up for performing the PCR technique on the DNA samples that were recovered on the crime scene, as well as from the victim and from various suspects. Okay, this is the laboratory setting, and we're going to perform PCR today. So we have pipette tips, we have pipetters, we have ingredients for PCR, and we have special PCR tubes for carrying out the reaction. And here we have a protocol that tells us the steps and as well the ingredients that are needed for the reaction. So obviously we'll need DNA, crime scene DNA, suspect DNA. We need master mix that contains the nucleotides, the buffer, the polymerase, the enzyme, and the primers. And so we're going to mix everything up inside the tubes here.
Just say it again.
So we're going to have to prepare the DNA. So we have the hair follicle inside here with some water. We've boiled it, and it contains the crime scene DNA, sorry. So that's the hair follicle here. And here we have suspect DNA that were obtained from various suspects. We have 4 of them. And now what we're going to do first thing is we're going to pipette using a pipetter here. We're going to take 20 microliters from each of these DNA samples and dispense them here accordingly in the tubes. I'm going to take DNA from suspect A. So I just press down, take the sample, and the sample is in my pipette tip, and I'm going to dispense it in the tube number 4. I get rid of the pipette tip because I don't want to cross. I'm ready to add the master mix, and very conveniently the master mix, which contains everything, is colored, so now I know I've added it to all my tubes. So for this I just need to add 20 microliters. It's a pre-mix. It's very easy. So I'm going to add and essentially I'm ready to go. What I need to do now is I have to close tube caps, press down, and I'm ready to put in the PCR machine. And the PCR, if you remember, is a thermocycler. See, all my tubes are ready. They all contain the mix. If you remember the PCR machine, it's a thermocycler. It increases and decreases the temperature automatically. So it will do the job for me, and then tomorrow morning I can come back and I can analyze my DNA samples. There you go. So here are my PCR tubes where I put all the ingredients. So now this is the thermocycler, and placing my PCR tubes in it, in those little slots. And I guess I'll have to close the lid, close it tight. And like I said previously, it's a thermocycler, so it will raise and lower the temperature automatically. So the first step is just boiling for 2 minutes, and then it's cycles that are repeated 34 times with 3 steps each. So the first step is denaturation, boiling. Second step, annealing. It's done at 52 degrees, and extension 72. And the timings are written there. So this is repeated 35 times, sorry. And then at the end, there's a final extension to make sure that all the fragments have maximum size. And all we need to do now is press run. Run crime scene, volume 40. Yeah, we added 2 times 20 microliters, so 40. okay, go ahead, and it does it automatically. Nicely shown here on the screen, the steps of the cycles.
Now, how are all these target sequences of different lengths analyzed and visualized? That's where we need another technique called agarose gel electrophoresis, which is always done in conjunction with PCR. This technique allows you to separate the different target sequences by size or by length and produces a unique pattern of bands, each band corresponding to a different target sequence. This pattern is called the DNA fingerprint, and each individual will have a unique DNA fingerprint. Okay, now let's explain the agarose gel electrophoresis technique. Visualize the agarose gel as being a slab of jello that is about 1 centimeter thick and has a shape of a rectangle. At one end of this slab, a series of wells are made and circled here, and it's in those wells, in those indentations in the slab of jello or agarose gel, that the DNA sample containing all those target sequences after PCR will be added, will be loaded. Now the DNA will enter the agarose gel and migrate through the agarose gel by a current that is applied. And typically DNA being negatively charged, it will move from the negative pole towards the positive pole. And it will produce, after staining, the DNA still needs to be stained, a series of bands. The bands that will appear at the top of the gel, they will be larger, that is closer to the well, and the ones that will be at the bottom of the gel, closer to the positive end, those will be smaller. So you get a pattern of bands called the DNA fingerprint, and let's say that this is the DNA fingerprint left at a crime scene. The procedure is repeated for victim's DNA, that is, victim's DNA is amplified, and then target sequences are separated by agarose gel electrophoresis, and you might obtain the following pattern. So clearly the crime scene DNA is different from that of the victim, so it might correspond to suspect. Let's say you have three suspects and you wish to amplify their DNA and separate the target sequence by agarose gel electrophoresis, and these would be the patterns of bands obtained. So there's suspect 1, suspect 2, and suspect 3. So 3 different fingerprints. So based on the fingerprints, the patterns of bands, you would say confidently that suspect number 1 is the perpetrator of this crime.
Okay, so the gel has been poured. It's a jello-like substance, a rectangular slab, and to pour it we need to prepare the bed, the gel bed, so we have to put tape. Now I'm removing the tape so that the current can pass through it once we place it in the chamber with a buffer. Removing the tape. And you can also notice that we have a comb here that essentially prepares the wells. Here we have the wells. Remove the comb, the wells, and that's where we're going to place the DNA, the PCR reactions. So we'll place it into the chamber with a buffer. Now we're ready to load. So I'm going to load 30 microliters, and I'm going to use the pipette tips again and change for every PCR reaction. We don't want to cross-contaminate. So here are my PCR tubes. Remove the caps. Caps gone, the tubes, the reactions, and so I'm going to load from every sample into a specific well. So the first well, well number 1, sample number 1. Now I'm ready to run, to pass the current, to do electrophoresis. I'm going to close the lid of the chamber up, and the setting is 200 volts for an hour, and all I need to do is press run, and it's running. Okay. So we know that the current is going because we have these bubbles that form at the electrodes, at the poles of the chamber. It's going to slowly migrate into the gel and separate according to size.
Solve the crime that was perpetrated earlier, we would say that Jerome on the left was killed by Ollie here on the right. He would be the crime and the suspect.